Assalamu alaikum. Today we'll be discussing the activation of the naive T cells briefly. This video will not be very long. So, the activation of the naive T cell from what we know until now, it happens because of the interactions between the T cell receptor and the MHC class 1 or class 2, depending on whether it's a naive helper T cell or a naive cytotoxic T cell. But that interaction alone between the MHC and the TCR is not enough to actually activate the T cell, the naive T cell, completely. It needs other co-stimulatory effects, and this is what we will be talking about mainly in this video. So starting off, one of the molecules that assist in the whole binding and the whole interactions are integrants. Integrants are small adhesion molecules. They allow basically for the cell to bind to its environment and get affected by its environment even better. So it assists in the transmitting of the signals between the cell and its environments. They are adhesion molecules, so they bring cells closer to its environment. In the case of naive T cells in particular, we're talking mainly about the lymphocyte function associated antigen. The LFA1 is present on these naive T cells. And this LFA1 is capable of binding strongly to the intracellular cell adhesion molecule ICAM1, which is present on the antigen presenting cells. So this antigen presenting cell has an integrin called ICAM1, and the naive T cell has this molecule called the lymphocyte function associated antigen. This LFA1 binds strongly to the ICAM1, which brings the two cells close to each other. And this allows for better and stronger singular transduction between the APC and the naive T cell to allow for stronger stimulation. Now, of course, these integrins are also helpful in the homing of the lymphocyte. So when a lymphocyte wants to know where to go, it can look for these ICAMs, for these integrins, and this will allow it to know where to go, basically. Now let's talk about the main bulk of this video, which is the co-stimulation of naive T cells. So here we have a naive helper T cell. And here we have an antigen-presenting cell with its MHC class 2 loading an antigen. And this is the T cell receptor. The naive helper T cell usually is expressing the CD28, and the antigen-presenting cell is usually expressing the CD40. And we will discuss them a bit in this exact same slide. So this binding is what we understood to actually activate the T cell, the naive T cell, the MHC molecule binding to the TCR. But this binding is actually not very concrete. It's not very strong. It needs something to stabilize it further. And what stabilizes this binding is the CD4 from the helper T cell. The CD4 binds to, if you remember, the beta 2 molecule of the MHC class 2, and this stabilizes the binding of the TCR to the MHC class 2. In the case of a naive cytotoxic T cell, this would be the CD8, if you remember. Now, with this, we have a stable binding between the TCR and the MHC, but this is not enough to actually get the full stimulatory effect. So once this binding happens, the helper T cell will try to activate the APC further so that the APC itself can activate the helper T cell. How does the helper T cell does that? It's by uh, expressing this CD40 ligand. The CD40 ligand, as the name suggests, it will bind to the CD40. Once this binding happens between the CD40 ligand and the CD40, the antigen presenting cell will be activated. And its activation will allow the APC to express this B7 molecule. This B7 molecule will bind to the CD28 molecule on the helper T cell. And this binding is the main co-stimulatory effect that will allow the helper T cell to actually be fully activated. Once this binding happens, the helper T cell is actually fully activated and will start expressing cytokine receptors. These cytokine receptors will basically be able to interact with the cytokines that are secreted by the antigen presenting cell, and this will activate the helper T cell further and further and further. So the main idea is that the binding of the MHC to the TCR is not enough by itself. It needs to be stabilized by either CD4 or CD8 depending on the type of T cell. And the helper T cell needs to get more co-stimulatory effects, mainly by the interactions between B7 and CD28. And this B7 is only expressed when the APC itself is activated via CD40 ligand to CD40 interactions. And the helper T cell, of course, always needs more stimulation via these cytokines and the cytokine receptor. So let's talk a bit about the B7 dependent regulation. And the B7 on the APC is not expressed in the absence of infection. So when there's no infection, we don't want the T cells to be activated, and so we don't express the B7. In the absence of B7, this naive T cell will not respond even after binding to the antigen on MHC. So the naive T cell cannot respond, cannot be fully stimulated unless there's also B7 co-stimulation. In the case of the absence of B7 co-stimulation, the naive T cell will go through anergy, which is basically its functions will actually depress rather than get activated. It will actually be deactivated and it will be harder for the T cell to activate itself afterwards. In the late stages of the immune response, we will find that the body actually wants to stop the inflammation. It wants to stop the immune response. How does it do that? It wants to lessen or decrease any stimulation of T cells. And this is done by basically this CD28, we don't want it to bind to B7 anymore. The B7 will stay and it will stay expressed on the APCs because the infection isn't resolved yet. But the infection or the inflammation itself, the immune response, is hurting the body. And so we want to stop it. So the B7, yes, it will stay, but the CD28 will actually be switched out or will be expressed less, while the CTLA4 will be expressed more on the T cell. So the T cell will start expressing the CTLA4 more than CD28. The CTLA4 has affinity for B7. It will bind to B7, and once it binds to B7, it will actually force the APC to remove the B7 from its membrane. And with this, it allows basically the body to stop lymphocyte stimulation by removing the B7, so there's no co-stimulation by B7, and every T cell will now go through anergy, and this will depress the immune reaction because we don't want it anymore. It's in the late stages of the immune response. We want it to stop. So this is basically the down regulation using the B7 CTLA4 interactions. Now, in the case, and there's another mechanism for downregulation, which is using the PD-1 molecule. This PD-1 molecule is expressed on all T cells, mainly on cytotoxic T cells, but it's also present on helper T cells. Okay. After antigen recognition, so after binding between MHC and TCR, we'll find that PD-1 is expressed on the T cell. Now, the ligand for PD-1 is present on all peripheral tissue. So all cells in the body will have this ligand. And it is upregulated during chronic inflammation and any time we want the immune response to stop. So once we want the immune response to stop, we will upregulate the PD-1 ligand. This upregulation of this PD-1 ligand, once it binds to the PD-1, it will activate phosphatases in the T cell. The TCR and CD28 signaling are actually kinase dependent, so they activate the cell using kinases, while the PD-1 will activate phosphatases, which will do the exact opposite of what the kinase will do, and so they will deactivate the cell entirely, stopping the immune reaction. And that is good in the case of a chronic inflammation or repeated antigen stimulation. But the problem comes when cells like tumor cells learn to express the PD-1 ligand more than normal. And now these tumor cells have this natural way of actually avoiding the cytotoxic T cells. Whenever the cytotoxic T cell wants to come and kill this tumor cell, they will bind to this PD-1 ligand, and this will deactivate the cytotoxic T cell, not allowing it to kill the tumor cell. And this is a problem, and this is one of the ways that tumor cells use to avoid the immune reaction. This is it basically for the naive T cell simulation, and these are the resources I used. Thank you, and see you next time.